Mr. Wiley, gonna get through membrane permeability tonight. Uh, Hopefully everybody stay with me now. All right, so we talk about membranes. Most important thing to think about, at least this is how my brain works anyway, is I think about like a screen. Okay, a membrane can't be letting just anything through. Okay, and we think about like a screen door and its purpose and why we use these things, right? We'll let air, breeze come through. That's what we want to let get into our houses, right? And we don't want the cat running out there and getting murked by some coyote, right? And we don't want these bees, these crane flies coming into our house all willy-nilly like, right? So we have these screens to let what we want come through and what we don't want to stay out. And that's really exactly how our membrane should work inside of our cells. We don't want toxins just, coming in, no issues, right? Without some kind of checkpoints. But we do want to have food come through. We want to be very selective about, items that are able to pass through in either direction. Okay? So the first thing that I want to get into here is what exactly can get through and how. All right? So we have here couple small non-polars. Okay, so what they're talking about, oxygen or carbon dioxide or nitrogen gas. This is our representative molecule here. How is it going to get through a cell? Obviously this is required for cell respiration, and this is what is going to be the byproduct of a lot of our processing of glucose, right? But how is it going to get into our cells? Okay, so here's how it goes. Right through those phospholipids. Okay, simple diffusion, right through the phospholipids. Okay, just to reiterate some previous information, hopefully everybody's with me, but these are all phospholipids. Okay, they're the major component of the cell membrane, and this is an embedded protein. Okay, so if it comes through here, it's going to be using a protein. If it comes through here, it is just going to be simple diffusion through the plasma membrane. Okay, and these small non-polar molecules are able to pass directly through the phospholipid bilayer. Okay, all right, so next up we got a steroid hormone. These tend to be very small, they're non-polar, and remember from our previous conversation that in here this is all non-polar in the middle where those fatty acids are. Okay, so if we want to get a steroid hormone, again, they cross directly through our plasma membrane. Okay, so testosterone, estrogen, any of our steroid hormones are going to be able to cause a change in the inside of the cell without having any activity with a membrane protein. They can cross directly through the phospholipids. All right, so how about a small polar uncharged molecule, like something like a water? Anybody want to take a guess? This part over here is polar, non-polar, so it should have some interaction, right? But these guys actually also can go directly through these phospholipids without using any kind of channel protein. And the only thing about this that I want you guys to feel comfortable with is that it typically is in much smaller amounts or less expedited than some of the other molecules we just discussed. Obviously when we talk about osmosis later, you guys will see that water it's not gonna be an issue for water to cross most cell membranes. All right, so what about like the primary food for most cells, the primary food source, which we break down in the mitochondria, which is glucose. Okay, so this is obviously a polar molecule, has a lot of hydroxyl functional groups sticking off it. It's gonna be polar. So a little bit larger, excuse me, a little bit larger, but these guys actually are going to have to utilize a protein. Okay, so in some way, shape, or form, polar molecules, especially the larger ones, are not going to get through the plasma membrane, through the phospholipids. They have to utilize a protein to help them. Now, if it's simple diffusion and they're utilizing this, of course, we call it facilitated diffusion, which we'll get into later. Otherwise, we might have to expend some energy to get this across, but again, that's a topic for another conversation. Okay, charged molecules as well, so we have like sodium or potassium. These things are also going to have to use that channel protein. Okay, so really important on the AP test, you guys are able to differentiate between a type of molecule, the molecule's properties, and what your prediction would be in terms of how this thing can interact with the cell, how it can get to the inside of that membrane. Okay, so I just want to clarify a couple different points that are utilized in different texts in different manners, but typically when I think of a channel protein is a type of transport protein. So this is kind of like an overarching descriptor, and then a channel protein is more specifically a type of transport protein, and in which the channel is open all the time. So I'm not expecting this protein to change shape, alter its conformation. It's possible, with a phosphorylation or a ligand binding, but for the most part, a leak channel or a channel protein is going to stay open, and it's going to enable specific particles, not any particles, specific particles from coming through that channel. And this channel is engineered in a way that it's only going to allow specific particles to pass through that channel. So again, channel protein open all the time. A carrier protein, on the other hand, okay, is going to be closed. It's going to have the ability to alter its overall conformation, and these guys typically can move in both directions in terms of the gradient. So you can have a situation where carrier protein is behaving passively, allowing things to go from high to low concentration, or it could be an active carrier protein, which is enabling things to go from a low to a higher concentration. Channel proteins typically are always going down the gradient, meaning that you have like more of a particle out here, and they're going to come across into the low concentration area. But a carrier protein can go in either direction. So typically we have a particle. This can be as simple as a phosphate group off of an ATP that is going to attach to this carrier protein, and that simple binding of that ligand or molecule in general is going to alter the shape of that carrier and open it up. Okay, and obviously if that opens up that protein, now I'm going to allow things to come through. And that's how I differentiate the different types of transport proteins. I hope I've made that clear. Both the channel and carrier proteins are both transport proteins. Primary difference, carriers are going to change shape. Carriers can go in either direction. Channel proteins maintain their conformation, and they only are going to go down the gradient from high to low concentration. So those are two different types of transport proteins. Additionally, in this part of the curriculum, we need to discuss the cell wall, which of course is a primary component found in mostly plant cells. So we're going to draw out a few things here, but what we have is a typical plant cell. Plant cells typically are depicted rectangularly. You have on the outside here a cell wall, on the inside here a plasma membrane, and then we have that central vacuole, which provides a lot of pressure that exerts outside or external pressure that it pushes out on the cell membrane, cell wall, and that's only if this vacuole is full with fluids. Okay, if it's a situation where the plant hasn't had enough water, hasn't rained in several days, the central vacuole will shrink up, and this pressure no longer exists. That pressure on a plant cell is called turgor pressure, and you've all seen plants wilt, and that's due to lack of pressure provided by that central vacuole. Okay, if you supply water to the plant, then the central vacuole fills back up, and that pressure pushes out on that cell wall. So right here, if we looked at that a little bit bigger, what we'd have is a phospholipid bilayer, and then just outside of that phospholipid bilayer, we'll have a series of structural carbohydrates. And this is typical of any cell wall. Structural carbohydrates are going to vary from cell to cell, but most cell walls are made of structural carbohydrate in some way, shape, or form. And interior to that cell wall is always the cell membrane. Okay? All right. So occasionally, not always, but occasionally you're going to have a little bit more to this cell wall, which I want to explain to you now. And also the cell wall components can be altered quite a bit depending on what type of cell we're talking about. Okay. So in this slide, I wanted to show you, a couple different things. Okay, so we have two cells that are next to each other; they're adjacent. Okay, and the cytosol you can see here is connected through this tube. This tube right here is called a plasmidesmata. You actually see that there's three of them here. Yeah, 1, 2, and 3. Okay, and so the plasmidesmata are going to allow fluid, cytosol, to to be exchanged. Now, if you're a plant, you're trying to transport water over long distances, sometimes up over 100 feet. It's beneficial to have these channels where we can transport water in a very quick way. Okay, the only problem with that sometimes is that plants, can have an infectious agent kind of go from one cell to the next through these plasma desmata. So they're really careful about the selective permeability. In other words, they're very selective about what gets through these layers, the cell wall, the cell membrane, and then eventually the cytosol. Okay, so when we look at this picture, I want to point out a couple other things besides the plasmodesmata. Typically, when new cells are formed, you have this middle lamella, which separates out the two cells. Okay, and then what you'd have is a primary cell wall here, and the secondary cell wall, which is interior to that. Okay, so this yellow, in this case, would be the secondary cell wall. The green is the primary cell wall. The middle lamella is in dark blue or almost like a purple color, I guess. And then this red is the cell membrane. Okay, and typically, you guys, the secondary cell wall is by far like the biggest, the chunkiest part of the cell membrane. Okay, so I hope everybody's with me there, and all of that is making sense. Okay, moving on. So the cell wall components are going to be primarily carbohydrates. In terms of plants, there are two main carbohydrates that are going to make up the cell wall. We have cellulose and pectin. Okay, and the pectin right here is kind of holding the larger cellulose fibers together. In addition, you have this lignin, which is an amino acid structure. It's not a carbohydrate. I don't want to confuse anybody. It's definitely an amino acid polymer, but a really big part of what makes plants, woody plants, very strong and rigid. Okay, so in a plant cell wall, mostly cellulose. That is the one that I would like everybody to try to keep in a memory bank. Okay, in terms of bacterial cell walls, we have a completely different complex. It's called peptidoglycan, and antibiotics, target this, prevent reproduction of that. And in terms of fungi, we have chitin. Okay, and so all of these are carbohydrates, and this is one of the primary differences between the cell walls of different species. And hopefully that makes sense to everybody. Again, with a plant, you have a very selective boundary on the outside of that plant. So we're not allowing just anything to enter into the cytosol and cause damage to cellular components in here. Okay, it's there's a vetting process. There's several boundaries that have to be crossed before we can get into and cause damage to the plant cell. Thank you very much for watching. Have a good day.